Hello students, myself Om Prakash Katte and you are welcome to Biology Reward. Today we are discussing the class 12th chapter Molecular Basis of Inheritance, and our topic is transforming principle and biochemical nature. Students, what is genetic material? Either RNA or DNA is a genetic material. So there are some experiment which shows that a DNA is a genetic material. So today we are discussing the very important experiment to prove that the DNA is a genetic material. Here topic, as I told you, transforming principle. Transformation. It is the process in which a genetic material of one organism or bacteria is passes or transferred toward the other bacteria. So this process is called the transformation. Genetic material of one bacteria, it's transferred to the other bacteria, and this is called transformation. So topic is transforming principle. How the transformation, genetic material of one bacteria transferred to other bacteria, and what is the chemical nature, that is biochemical nature of genetic material. So that genetic material is protein or DNA that we have to study now. Students, Frederick Griffith. This Frederick Griffith, or commonly we are used to say Griffith experiment. So Frederick Griffith performed the experiment, and it is called Frederick Griffith experiment in 1928 to show that DNA as a genetic material. It means DNA is a genetic material. But he proved, Griffith experimentally proved DNA is a genetic material. For this experiment, he worked on experiment on the pneumonia causing bacteria. So for this experiment, he worked on the bacteria which causes the pneumonia. Name of bacteria, Streptococcus pneumoniae. So in this experiment, he took the pneumonia causing bacteria, Streptococcus pneumoniae, and he observed, he witnessed the miraculous transformation in bacteria. Okay, so he observed that in pneumonia causing bacteria, Streptococcus pneumoniae, he observed that the transformation occurs in this bacteria and they change in physical form, and their physical forms are getting changed. So experimentally, he observed that the transformation occur in bacteria and this form of bacteria get physically changed. So they change in the physical form. So he worked on this Streptococcus pneumoniae, and what he observed? He observed that transformation occur in bacteria and they change its physical form. Now in Streptococcus pneumoniae, he observed two states. If the colonies of the bacteria are grown on the plate, so there are two types of varieties of bacteria, or this we used to say strain. So in Streptococcus pneumoniae, first strain is strain S. And strain, it means variety S of the pneumonia. So this strain S is what? Virulent. Virulent, it means the strain S, it causes pneumonia. Virulent, it means causes disease. So the strain S is a virulent, it means it causes disease or pneumonia, and it has smooth surface. Strain S is for the smooth only. So the strain S is having the virulent, it causes the pneumonia, and it has smooth surface. Why the smooth surface? Due to polysaccharides coat. This type of strain of bacteria, they are causing the diseases, pneumonia, and they have smooth surface, and this smooth surface is called the polysaccharide coat. So if I will make the diagram, this is the bacteria, and outside the bacteria, this is the polysaccharide covering. So this type of smooth surface, it means smooth surface is there, and this stain of the Streptococcus pneumoniae bacteria is called the stain as stain. So it was the first variety. Another type of stain also observed in the Streptococcus pneumoniae, and it is called rough stain or stain R. So stain S, virulent, smooth surface due to polysaccharide, but stain R means rough. And this type of stain of bacteria non-virulent, they do not cause the pneumonia. They do not cause the pneumonia, and they have rough surface. And rough surface is due to absence of polysaccharide coat. So in this, they have rough surface due to absence in polysaccharide coat. So we should remember there are two strains of the Streptococcus pneumoniae. Strain A, strain S, it is virulent, it causes the pneumonia and smooth surface due to the polysaccharide coat. But strain R is a non-virulent, it will do not cause the disease, pneumonia. They have rough surface, and in such cases this coat does not occur. So this coat is not occur, so their surface, this surface part is the rough surface, and that's why it is called stain R. So we have two stain of the bacteria. One stain is called stain S, which is virulent and smooth surface, and stain R, non-virulent and rough surface. So he observed these two stains. Now what he performed? He performed an experiment. What was this? First one step. S stains, which is the virulent stain, is injected to the mice. Mouse everybody is knowing. Rat we are knowing. So S stains he injected to the mice. So what he observed? So S stain is the virulent stain. So the mice suffer from the pneumonia and lead to death. It means S stain when injected to the mice, they die. Why they die? Due to pneumonia, because S stain is the virulent stain, and due to the virulent stain, disease-causing stain, they suffer from the pneumonia and lead to death. Second experiment, now R stain injected to the mice. First S stain, now R stain injected to the R stain is non-virulent stain, we do not cause the disease. So now R stain is injected to the mice. So what happened? Disease is not caused and mice are alive. They do not die. So they alive. R stain is injected to the mice, then mice are alive or live, do not die. Because this is non-virulent stain, do not cause the disease. Now third experiment, S stain is heated. The first S stain, it heated. So when S stain is heated, what happened now? The bacteria will be died due to the heating. So S stain is heated. Now what happened? And it injected to the mice. It is heated and injected to the mice. What was the result? Again the mice life. The reason is what? Bacteria are killed due to the heating, and that's why the mice do not suffer from the pneumonia, and the mice are live. S stain heated and injected to the mice, then they are live because they do not suffer from the pneumonia. But in fourth experiment, S stain get heated and they mix with the live R stain. S stain is get heated and they mix with the R stain. R stain is alive. Then this mixture of both, S stain heated plus R stain, mixture of both is injected to the mice. So what result came? Again the mice died. You see, S stain heated and injected to the mice are live. But now S stain heated and they mix with the R stain, rough stain, Now injected to the mice died. So why does this happen? S10, I told you, here the bacteria are killed, and that's why this they do not cause the pneumonia. And S10 heated is injected to the mice, then mice are alive. But S10 heated now mixed with the R10, dimustane, then injected to the mice die. It means the mice are suffering from the pneumonia. So the Griffith said that, conclusion, R strain bacteria transformed. What is transformed? Because this R is the non-virulent, but here when the S10 heated is mixed with the R10, it's a mice suffer from pneumonia. It means R10 bacteria, this bacteria transformed. Which one bacteria is transformed? R10. Which is changed? R10 bacteria. R10 bacteria transformed by heat killed S10 bacteria. Somehow the S10 bacteria changed the R10 bacteria. S10 is changed to R10, and that's why we say R10 bacteria get transformed by heat killed S10 bacteria. So, second conclusion. Some genetic material, he said that some genetic material transfer from S10 to the R strain, and R strain bacteria get transformed. What does it mean? Nature of R strain bacteria is changed. Why change? Because some genetic material of S10 is gone to R strain. Because here S10 you are hitting, then no pneumonia caused. Mice are alive. But here what happened? S10 heated and it mixed with the R10. So something from the S10 goes to the R10. It means R10 get transformed. And second statement said that some genetic material transfer from the S10 to R10. And that's why this R10 become virulent. It means it causes the pneumonia or disease. So this experiment shows that some genetic material of S10 is get transferred to R10, and this I told you it is transformation. This is the process. Genetic material of one bacteria is transferred to other bacteria. So here what happened? R is non-virulent strain, but when S10 heated mixed with the R10, now it becomes the virulent. And when it is injected to the might lead to death. So it means R10 bacteria is transformed and transformed by S10 bacteria. Second, genetic material of S10 is transformed to the R10 bacteria. So in this picture also you can see all four experiments. First one, S10 bacteria when injected to the mice, then they die. When R10 bacteria injected to the mice, they are live because pneumonia is not caused. But when S10 is heated and it is injected to mice, then the mice these mice do not die. It means they are live. But fourth case you will see S10 heated is mixed with the R10 and now injected to the mice. Then mice are suffering from the pneumonia and lead to the death. And overall by this experiment it shows that the R10 get transformed by S10. It means the genetic material of S10 is transferred to the R10, and this is overall about the Griffith experiment. This experiments many times come in the examination. So examination point of view, it is very, very important question, Griffith experiment. But in this Griffith, do not explain about which material is transferred. What is the genetic material? So he not explain biochemical nature of the genetic material. He do not explain the genetic material is DNA, or this is RNA, or this is protein. So he only said that genetic material is transferred from the one bacteria to other bacteria. So not explain biochemical nature. So the biochemical nature of the Griffith experiment, biochemical nature of genetic material. Biochemical nature of genetic material. It is explained by the three other scientists. Oswald Avery, MacLeod and McCarthy. While they were doing the experiment on the Griffith experiment, similarly, Griffith-like of experiment they performed. Let's say Oswald Avery, MacLeod, McCarthy and these three scientists, they give the biochemical nature of genetic material. What is actually transformed? It is DNA or it is protein. So, first they have purified protein, RNA and DNA from heat-killed XII bacteria. As I was talking, from XII heated bacteria, they have purified some protein, they have isolated some proteins, RNA and DNA. Okay, so first was there, all these three they purified proteins, RNA and DNA from heat-killed as bacteria. From heat-killed bacteria only, some genetic material get transferred to XII. So they purified the protein, they have isolated protein, RNA and DNA from heat-killed as bacteria. Now what happened? Heat-killed XII bacteria, first experiment, heat-killed XII bacteria they have taken with live XII. So there is a heat-killed XII plus live XII and they mix the proteases enzyme. Proteases enzyme, this helps to digest the protein. So heat-killed XII bacteria, live XII bacteria and they mix proteases protein. So what happened? Whatever material is there, that protein get digested. So when protein get digested, what happened? Transforms R10 to S10. In such cases, if protease enzymes are mixed, so protein get digested, but what happened? This transformation process occur. It means R10 is changing to S10. It means process of transformation is not affected. When proteases is added, the process of transformation is not affected. They are transformed R10 to S10. It means R10 become virulence 10. So here proteases digest the protein. It means protein is not the genetic material. Second, in such cases, heat-killed XII bacteria mixed with the live R10 and RNase enzyme. It was proteases. RNase enzyme is used to digest the RNA. So when it is added in this suspension or solution, here solution is of heat-killed XII bacteria, live R10 bacteria and RNase. So RNase will digest the RNA molecule. So when RNA molecule is also digested, what happens? Transform R10 to S10. It means it is also transformation process is continuing. Here proteases you are added, so transformation is continuing because R transform the R to S. If RNase you are adding, transformation is also continuing, and this R10 is act as a assistant. Third experiment, heat-killed XII bacteria, same, live XII bacteria, and now added the DNase. DNA is used for the digestion of DNA molecule. So when DNA is added, now what happens? Unable to transform R10 to S10. Now in these two cases it was virulent because R was changing to S. Here also R was changing to S. But when DNA is added, now R is not changing to S. It means unable to transform R10 to S. It means somewhere the DNA is digesting the DNase enzyme is digesting the DNA. So when DNA is get digested, transformation not occur. Here you are adding proteases, which digest the protein, but transformation occur. Here you are adding RNAs, it digests the RNA, and that transformation also occur. But when you are adding now DNA enzyme, then unable to transfer, it means transformation is not occur. Genetic material is not transformed, and that's why here DNA is adding, digest the DNA molecule, and when DNA molecule get digested, no transformation is occur. So what is conclusion? Conclusion brought by these three scientists is, it is proteins and RNA are heat-killed substance are not genetic material. Because this protein and RNA are digested, but transformation is occurs. It means they are not the genetic material. But if RNA is added, then transformation is brought. It means the main material, DNA is a genetic material, is proved, because DNA digest the DNA. So when DNA molecule is digested, now no transformation is occur, and that's why in transforming principle of Griffith said that the genetic material transfer from one organism to other bacteria, one bacteria to other bacteria, and biochemical nature, it proved that the DNA is genetic material, not protein and not RNA. So again, in brief, you understand, Griffith experiment proved that the genetic material is DNA and it gets transformed from one bacteria to other bacteria. But in biochemical nature, here this shows that the protein and RNA are not a genetic material and DNA is a genetic material get transferred from one bacteria to other